Hello, I am Sam Smith, and this is Sam's MCAT Basics. This podcast covers the most important topics put out on the AAMC MCAT each year, and I determined this list by going through all the official practice materials that the AAMC puts out, and also some third-party practice material, and just put together this big list of topics that consistently kept on showing up, and ranked those topics and made podcasts out of it. This podcast covers amino acids, which are a very important topic. You'll probably see about 10% of the questions asked will be something regarding amino acids. And so amino acids will show up on 2 out of the 4 MCAT sections. It'll show up on the chemical and physical foundations of biological systems, and it'll also show up on biological and biochemical foundations of living systems. So I hope this podcast helps you in your studying. Enjoy. So the first thing I'm going to go over is naming and also the structure and the abbreviations of the amino acids. But before I start all that, there's one little piece of information you should probably know for the MCAT, and that is that the average molecular weight of an amino acid is 110 kilodaltons. Just keep that fact in your back pocket for use. All right, so on the test, you're going to be given the amino acids usually in their 1-letter code abbreviation. So don't pay too much attention to the 3-letter codes. Just really focus on the single-letter codes. And so most of these 1-letter codes correspond to the first letter of the amino acid name. For example, alanine is A, valine is V, glycine is G. There's some different weird ones, though, where that's not the case, and I'm just going to go over those to save a little bit of time. So the first is E, which is glutamate. Next there's Q, which is glutamine. Next is K, which is lysine. Next is F, which is phenylalanine. Y, which is tyrosine. R, which is arginine. N, which is asparagine. Then you have D, which is aspartate. W, which is tryptophan, and then sometimes you'll see an X, and that's some other kind of amino acid or some unknown amino acid. So real quickly, I'm just going to go over how I can kind of remember these. So W, I just remember tryptophan, like you talk a little bit with a list, tryptophan. Let's see here then, tyrosine, that's the second letter, and that is a Y. What are the other ones? Phenylalanine, easy to remember. Like it's PH, but it looks like an F, or it sounds like an F. So the other ones, you can kind of develop your own technique. I always remember that a nutritional supplement sometimes that I'll take is glutamine, and that's Q. And so without a question, I'm always going to take glutamine. And then it's also easy to take glutamate, which is just a similar form. So I remember that is E. And then you can remember that D and E are right next to each other. That's glutamate and aspartate. And then in arginine, you can do the good old pirate trick, the arginine. So you just remember it's R. And there's a few. So you can develop kind of your own little systems of ways to remember them. You might still obviously have them memorized from biochemistry, but if not, kind of figure out your own ways or use mine, whatever. And I could definitely go over the structure of each. However, I'm not going to do that. I don't think that's going to be very helpful just to describe the structure, but definitely make sure you know each structure. There's definitely questions that I've seen on the test where it'll literally just show you a picture of an amino acid and essentially say what amino acid is this, or usually in a more convoluted way it will say something like what amino acid is this, just based on the structure. And these amino acids can be grouped into hydrophobic, hydrophilic, or polar and nonpolar. And so each amino acid has a general structure in which it has an amino group and a carboxylic group with a R group coming off the side. And so that's why it's called an amino acid. And the R group in the amino acid gives it its hydrophilic or hydrophobic properties. And so in another way they can be characterized is either charged or non-charged. So I'll go over what all these mean and which ones are which. So the hydrophilic amino acids are the amino acids with polar side chains. And so these are side chains that contain either hydroxyl groups, which are OH group, and amine group, NH3 group, or a carboxylic acid group, or lastly, they contain an amide group. And so an amide group is essentially carboxylic acid, but instead of the OH attached to the end, it's got an NH2 group, which is an amine group. And so the hydroxyl-containing amino acids, again, these are hydrophilic, are serine, threonine, and tyrosine. The NH3 or amine-containing groups are histidine, arginine, and lysine. And then moving on, the carboxylic acid containing R groups are glutamate and aspartate. And then lastly, the amide containing amino acids are glutamine and asparginine. And these groups are called hydrophilic, which comes from water loving because they can form hydrogen bonds with water, and they interact with water. They love water very much. Moving on now, the hydrophobic amino acids are amino acids with non-polar side chains. And so these contain hydrocarbons, which are just hydrogen and carbon. And so these are leucine, isoleucine, alanine, phenylalanine, tryptophan, methionine, which does contain a sulfur, but that sulfur cannot hydrogen bond water and is therefore non-polar or hydrophobic. And so again, these compounds or these amino acids with the hydrophobic R groups cannot hydrogen bond with water and therefore fear it, hydrophobic fearing water. Next, I'm going to talk about which amino acids are charged. And so this is particularly important. I think you're bound to see a question in which it gives you a sequence of letter codes and it says, give me the net charge for this peptide. And usually every single instance I've seen is it's going to be at physiological pH. And so I'll talk about what exactly that means. And those should be give me's. It's a really easy question to solve. So the charged amino acids are the ones that are able to donate or accept a proton or a hydrogen ion. And so the acidic groups, which are able to donate protons, are aspartate and glutamate. And the basic amino acids are lysine, arginine, and histidine. And these are able to accept protons and become positively charged. And so, as I said before, the questions you're going to see are going to ask you at physiological pH, what's the charge of this peptide? And so there's four amino acids that are charged at physiological pH. I'm going to go over these right now. The first is lysine. And so, as if you can recall, I said lysine is a basic amino acid. So that means that at physiological pH, it accepts a hydrogen ion. It has a positive charge. So lysine, physiological pH, positive charge. The next amino acid is arginine. And so again, I was talking about how that is one of the basic amino acids. And so at physiological pH, it's going to be a +1. And then you have glutamine, which is going to be which is an acid proton donor and is going to have a -1 charge at physiological pH. And lastly, you have aspartate, which is also going to be a -1 charge, just like glutamate. And so how I solve these problems is I remember the mnemonic street cred with a K. And so cred, K-R-E-D, those are the 4 amino acids that are charged at physiological pH. And what I would do is I would go through on the question, I'd write down the sequence of one-letter codes that it gives, and I'd circle each one that was K, R, E, and D, and then I'd go through and you just mark the charge of each, add them up, And there you go. That's your total overall charge. And now that I'm done talking about structure and naming of the amino acids, I'm going to move on and talk a little bit about mutations. So on the MCAT, mutations will be presented in a certain notation. And if you have done any kind of lab research on amino acids or done lab research on different proteins, you're likely to have seen this. However, if not, you may not have seen this. It's going to be presented in the form of a letter, a number, and then another letter. So it's going to give the first letter corresponds to the amino acid that is normally present within the protein. The number is going to give you the position at which this mutation occurs. And then the third component, which is the second letter, is going to be the amino acid that is substituted in. So let me give you a quick example. So, for example, K285D, that would mean that lysine, K, at the 285th amino acid in the protein sequence has been mutated to a glutamate, which is D. It's pretty easy, but it's important to go over. And mutations can affect a lot of different parts of a protein. I'm going to go over a few different ways. This is definitely another very common question on the MCAT. So the first way that a mutation can affect a protein is when this mutation affects the substrate pocket for an enzyme. And so one of my podcasts is actually going to be just on enzymes. So all you need to know right now, if you don't know, is that a substrate pocket is where a molecule will bind to the enzyme, and then the enzyme itself catalyzes a reaction which spits out a product. So the substrate pocket is where that reactant binds. And so the substrate pocket itself can be involved in a number of different things. It can be involved in stabilizing the substrate and keeping it in the substrate pocket, or it can be even involved in the reaction itself to convert that reactant to the product. And all these functions of the substrate pocket are based on its amino acid makeup. And so a lot of times you'll have these different R groups in the substrate pocket which perform many different functions. And so I'm just going to list off a few of them for you. The first is they can act as acids or bases, which is a pretty important factor, especially for reactions. It can also act, these R groups that are in the substrate pocket can act as nucleophiles. They can stabilize the transition state of a reaction. It can activate water by deprotonating water. It can activate some cofactor. It can act as a primer. It can form a radical. And so that's just a few examples, and these R groups are very important. And so when you have a mutation, that can really affect the functionality of an enzyme. And so I'm going to go over a little bit of how that happens. And so a mutation that basically changes an amino acid in the active site from polar charged to nonpolar is likely to affect the enzyme's function. So you can imagine an OH group in the substrate pocket, right? And so that's, say that acts as a water deprotonator. And so if that were to be mutated, let's call it a tyrosine. Let's say that tyrosine was mutated then to an alanine, and now you're losing that function there, and your enzyme might not be functional at all. However, the exact reverse could be true too. So let's say you have an active site tyrosine, like I was just talking about, that OH functional group sticking in the inside of that substrate pocket, and you have a mutation in which you mutate that tyrosine to an alanine, but you then measure the activity of that protein and nothing changed. So what exactly does that mean? Well, that probably means that amino acid did not have much to do with catalyzing the reaction. So maybe that has to do more with the structure of the actual pocket itself, and substituting those two amino acids didn't have anything to do with changing the shape of the substrate pocket. But that's just evidence that OH group, originally from tyrosine, may not have had anything to do with catalyzing the actual reaction. It's important to note that cysteine is another amino acid that can act as an acid or base, and that it acts at a lot of the active sites of proteases, which cleave proteins into smaller fragments. It's important to note that non-polar amino acids do play a role in the substrate pocket as well, and so they can stabilize different molecules while they're in the substrate pocket. To give you an example, phenylalanine, which has that big phenyl ring, which is a benzene ring, can interact with other non-polar amino acid residues via van der Waals interactions, also called hydrophobic interactions. Other big bulky groups with lots of carbon and hydrogen can interact with these phenylalanines and kind of stabilize this reactant in the substrate pocket. And outside the substrate pocket, mutations can also kind of mess with the protein, and we'll get into that next of how these mutations can affect stuff outside of the substrate pocket of a particular protein. All right, so mutations can also affect the overall three-dimensional structure of the protein itself, not just that of the substrate pocket. So next, I'm going to talk about how amino acids play a role in determining the overall three-dimensional structure of proteins. You may have heard this called the tertiary structure of a protein. So just know when I'm referring to the overall structure, I'm talking about the tertiary structure. Before I get into the mutations that are going to affect protein tertiary structure, you should know that there's a few very important amino acids in determining this overall structure of a protein. So the first is cysteine residues, and a residue in molecular biology refers to a specific monomer within a polymer chain. So in this case, it is a single amino acid. And so these cysteine residues can form disulfide bridges that basically link covalently different parts of the peptide together. And then it's also important to note that hydrophobic amino acids will tend to be buried at the center of a protein, while hydrophilic amino acids will be located on the exterior of the protein in contact with the aqueous solution surrounding that protein. And this is simply because the hydrophilic amino acid residues prefer aqueous solution. And lastly, salt bridges can also form that support protein structure. And so salt bridges are just a link between electrically charged acidic and basic groups, especially on different parts of a large protein. And so mutations can affect these in a few different ways. Obviously, if you have a salt bridge in your normal enzyme and then you mutate an acid that's away from the substrate pocket, but you mutate this amino acid from a basic group now to an acidic group, now what's going to happen? So now you have an acidic group and an acidic group that this mutated basic amino acid used to be attracted to, but now you have like charges, and those are going to repel, and that is really going to do a number on the stability of your protein. So as you heat it, this thing's gonna break right down. And I'm gonna give you a quick example of this concept, kind of and tie everything together. So you have pepsinogen, which is a peptidase in the stomach, helps break down peptides, makes them easier to digest. So let's say that this peptide undergoes a K25D mutation. This is the notation you're going to see on the MCAT. And so to decode that, you have K, that's lysine. So you're going to say lysine, the lysine in pepsinogen at the 25th amino acid position was mutated to a aspartate, which is D. And let's say that because of this mutation, you know that when this enzyme is heated, it becomes dysfunctional at a much lower temperature than the wild type or the non-mutated enzyme.
And so it's going to say, okay, what does this mean exactly? And so if you think about it, the K or lysine has a positive charge at physiological pH, and that should make you think, okay, maybe salt bridge. And so now let's look, what did it get mutated to? It got mutated to D-aspartate, which has a negative charge at physiological pH.
So this specific amino acid or specific residue within the protein went from a positive charge to a negative charge. And at this point, you can kind of take all the information they've given you, put it together. You know that you have a change in charge. You know that your enzyme became less stable. And you can start to conclude that the amino acid at position 25 was involved with a salt bridge. Furthermore, what's likely going on is
Two like charges, you have a minus from aspartate and you have a minus presumably from some other residue in the amino acid.
and these like charges repel each other, and your protein now is less stable. Boom.
And it's also important to note that mutations in cysteine residues can also affect protein folding and enzyme activity.
These, I like to call what's wrong with my enzyme problems, come up quite a bit on the MCAT, and they're kind of fun, make you feel a little bit like Sherlock Holmes, and a little investigative. They'll give you that little aha feeling when you figure them out. But just keep in mind these come up quite a bit, and I'll talk a little bit more about these kind of problems on the enzymes podcast.
And so
If your protein or your enzyme has some kind of disulfide bridges, then if that cysteine becomes mutated and you can no longer form that bridge again, you're going to have a protein that's less stable. And so a lot of times on the MCAT, how stability is measured is protein is heated up or an enzyme is heated up, and its activity is looked at as a function of temperature. And so an enzyme that's become mutated will typically degrade at a lower temperature than, say, the wild type enzyme that doesn't have certain mutations, or an enzyme that has mutations that do not affect its overall protein stability or folding. And so this might be one of the shorter podcasts, but is really probably one of the most important. You should know a few main topics on the amino acids. Have their structure memorized, have the abbreviations memorized, memorize the physiological charge of each, as well as know which ones are hydrophobic, which ones are hydrophilic. Look over mutations. Look into a little bit on protein folding and protein stability, and you should be set for the MCAT. These questions really should be the give me ones. You shouldn't miss any of these ones. Learn these few main topics on them and then just apply it to the rest. And I hope you learned and hope you listen to the next one. I think it's going to be on acids and bases, so it'll be a good one. Thanks for listening. So the last segment I want to do is the MCAT advice of the day. So the advice I have is to schedule your exam early. So, maybe within the first few weeks of studying, as you get an idea of how much you're going to need to study, figure that out, and then set your test date. I believe that this helps you kind of get focused, get ready for the test. Whereas if you don't set it, you're kind of like, I could study today, I couldn't, I might take the test 5 months down the road, or 3 months, whatever. You don't really have a set time. But when you give yourself a set time, it just helps you kind of focus for that. I know too many people that don't set their date and then just kind of half-ass study for a certain period of time, and a year later they still haven't taken the test. I don't think that's the most efficient way to study. If you're looking for an efficient way to study, set your test date and then stick to it, take the test, study hard. Come on, Murph. I'm trying to podcast over here, dude.